Okay, let's take a look at some of the student questions during the break. Uh, so last lecture we went over this so-called out of Africa theory. So maybe obviously someone was shocked uh, to know that we are all descendant of this original those migrants from Africa. Yes, we are. Yeah. So but that's not that doesn't mean that all of the humans lived in Africa all together moved out of this Africa so that no one is left ever since then. No, that is not the case. So still there were the majority of the population in Africa, original the native African, so to speak, remained there, and so currently, what you see in the descendants of the original African, that's why there are more genetic diversity can be found in Africa than the rest of the area human population, because the rest of the population are actually established from very small number of immigrants, actually. Immigrant, should I say. So in terms of number wise, they were much fewer than those who remained in in Africa. But speaking of all those human outside of the Africa now are actually originally the descendants of those first small group of humans migrated out of Africa. So then the question is, then what has made this different skin color and all together? So that is actually mind-boggling question. So if we all were derived from the African, originally we should have had darker skin color and some of the particular fixed outside appearance. But then how come we have, like, for example, the white Caucasian, for example, Scandinavians and Asians, and well, what makes this racial differences? Actually, I remember I mentioned briefly about this before. Um, the human genome, so-called Human Genome Project, which was the 10-year long, very expensive project, pouring billions of dollars of U.S. taxpayers mainly, revealed one standard whole entire DNA sequence of a human, exemplary. It took 10 years. But once that has completed, this Human Genome Project, scientists were so excited about the prospect of a next step. Once you have figured out one standard genome, whole genome sequenced, and the next step is now compare. Let's compare. So this is a standard genome. Now, would there be any difference in terms of this DNA sequence among different groups of races, like blacks? There is some specific genes which is only typical characteristic of blacks, whereas there is some specific DNA sequences which only present in the white and like the Asian too. So that's the next step. They rushed into such project immediately, and the end result was, oops, nothing. There is no difference among different races in terms of the DNA sequences. Then what happened? I mean, obviously everybody can tell that there is a difference in terms of outside appearance, like blacks, whites, and Asians. And how come they don't show such corresponding differences at the DNA level, DNA sequence level. So the question is this an example of heritable acquired trait? The other probably terminology describing this is what? Anyone remember? Epigenetics. Genes, the trait, something not involving in DNA sequence changes is by definition is epigenetics. Yeah, I mean that's probably scientists had to agree on the racial differences are in generally there may be a little very minor typical trait that is actually shown in terms of DNA sequence level differences, but overall, these racial differences, at least physically, outside the external racial differences, is something due to this, like example, acquired trait due to the different environment that somehow happens to be passed on to the next generation, transmitted. So this question is very similar. Why do humans and their ancestors look similar? Yeah, similar enough. But animals like birds and their ancestors, maybe dinosaurs, look very different. I'm afraid you are talking about apples and oranges. Birds, you said birds, but each individual bird is independent, separate species. So if you apply the same principle to these humans, then you have to bring in many different all kinds of different animals and talk about these common ancestors. So that's why. So if you think about these all different birds as yeah, this is a really easy mistake. When we think about plants, plants are all plants. So we sometimes forget that each different plants are like independent, their own separate species, like human and chimpanzees. Science is not a truth, and scientists themselves say nothing is 100% true. Yeah, that's right. Science, nobody says that science is a true or a fact or anything. So there is always, that is the beauty of the science, that anytime it has a chance of being not true, can be corrected. So all this science information, the factors, is a temporary factual information or theory even, is always ready for like another improvement or being corrected later on. So that's the, personally I believe that's the beauty of science. And then this lady specifically mentioned the possibility of this zygotic barrier. These days, actually, one of the strong mechanism keeping two separate species from interbreeding, so that like providing some of the hybrid, is this zygotic barrier, probably. But as we can easily imagine, this zygotic barrier cannot be 100% bulletproof, obviously. Right? Sometimes some of the escape or mistake, whatever, some exception can happen, right? And the very small, even if it's a very small, rare possibility, but still that possibility we cannot rule out, right? True. Then can it lead to some kind of a hybrid? But if you think, expand your imagination, hybrid can become as drastic as some kind of a chimera. Yeah, I like that. Yes, that's true. Chimera. Like, for example, a hybrid between human and fish could be a mermaid, or This one, I'm not really sure what she meant, but this one reminds me of some other possibility of there can be a human and a fish hybrid, so that like human having a gill in their ears, so that human doesn't have to be drowned anymore, because you can breathe, still breathe through the gills in the water, and then outside of the water you can breathe in the lungs. Some salamander actually some salamander have this gill still in behind in their ears. So many different types of this weird looking hybrid or chimera possibility. But to have this, a lot of things has to fall into correct combination. So in terms of this possibility, still, unfortunately, it's extremely rare, especially in nature. So the answer is of course, we cannot rule out. Yeah. After all, each of our existence is such a really rare, exceptionally rare chance event, but still we exist as we are. So we cannot, there is no way we can completely, this is out of the question, rule out, but still in reality, this possibility is still is very extremely low. Uh, yeah, I feel for you. This is not a lecture, but last week's the lecture was really difficult for some of you. So I'm not sure like how she felt about this today's lecture. Does she feel better or even worse? So my sympathy, my whole true sympathy for you. But the only thing I can say is I will try my best to make it easier. Easier to understand is all I can say, or I can promise. Is the difference in the number of chromosomes also a reason why different species of animals cannot breed? Yes, exactly. First off, the difference in the chromosome is the first barrier. Because if you have a different chromosome and when you generate with that different chromosome number, think about that. With such a differences in chromosome number, diploid number, if you generate the haploid chromosome number, it wouldn't match. Like for example, 12 versus 13. You don't have any partner. At least one chromosome will not have any partner. When they have this, once again, restore the diploid status through fertilization. Another more important reason behind this is the concept of this homologous chromosome, as we have uncovered last week's last lecture. This is not merely about the number-wise chromosome partnership, but by definition, this homologous chromosome is, let's say, if this chromosome is homologous to another chromosome, that means physically, appearance, they are identical in terms of their appearance, but also more importantly, the spot-on comparison. This exactly same location between these two homologous chromosomes must have the same gene. I'm not saying the same trait, but at least the same gene. Like if this area on this location of this chromosome has a gene for making eye color, okay, and exactly the corresponding location of this homologous chromosome also should have exactly the gene responsible for this eye color. Which eye color doesn't matter. It could be black eye or brown eye, but at least should be eye color. Same thing. So along these two homologous chromosomes, kind of genes located alongside of these two homologous chromosomes should be the same. That's the definition of a homologous chromosome. And that is the reason why they pair each other during this cell division, the meiosis, the sexual cell division. And that's the basis of this, whether certain fertilization should be like viable, or certain combination of this sperm and egg is non-viable, so like produce only sterile individual, etc. Okay? So there is much more than mere chromosome number exist for this, the reason for this not being able to interbreed. Well, what can I say? If you inherit much less gene from Neanderthals, then would you be happier? Personally, I don't think so, because happiness is not something about like what kind of gene you inherited, but how you think. How you think your particular current circumstances is what make you like have different degree of happiness. So although I can understand where this particular the person who had this question is going, but answer is I don't think so. And how do you propagate the seedless watermelon? Because originally to create the seedless watermelon, you have to have so to speak normal diploid watermelon crossed with doubled up watermelon, then because each will generate N and 2N and combine have a triploid, and this will be a watermelon, because once you have this triploid, you are not going to be able to produce any seed because you cannot produce any viable sperm or eggs. Okay. So that's how you create this seedless watermelon. But if you think about this, then do you have to do this every time? Like every year you have to cross this, prepare the new set, and then try to cross manually is the question. Isn't it too cumbersome? Yes, it is. But there's no other way. You have to. That's why probably seedless watermelon is relatively rare and more expensive. And yeah, there is like a cheat key exists. Okay, so if you really don't like every year going through this, then there is another way of something called cutting. It is a type of asexual reproduction. You put some stack of watermelon, seedless watermelon, then cut it out, and then plant them, and then it will grow as an individual. It is really a common thing for many plants. So if you don't like crossing these two every year, you can do this once you have established a seedless watermelon, and then from them you can just keep doing this cutting. But this cutting also is not really an effortless. So either way, you choose. It's depends on your preferences. Is it completely impossible to degenerate to evolve for the better? I'm not really quite sure what this particular question means. Degenerate to evolve for the better? First, evolve for the better or worse is not, it is something very artificial definition. Superficially or like temporarily, you may actually consider some type of evolution as a better evolution, but later on it may turn out to be it's not really as bad as or as good as you may have originally thought. So better evolution, worse evolution, defining this thing like artificially is something a little risky attempt. And degenerate probably means like the opposite word of progressing, improvement. So making, you may think that actually in order to become better in terms of evolution, you always have to be more complex, sophisticated. No, it is not. Sometimes evolution doesn't care. Evolution doesn't actually care the outcome. So it is only something that that particular times, particular external outside environment will dictate or to dictate what you may have become. So as a result, you may become a much simpler organism to adapt, to show higher adaptability, or vice versa. So I said it is not completely impossible. Sometimes you can become much simpler in terms of like outside or in the function wise, and if that's what evolution in terms of natural selection required for you to survive. So okay, so that's so. Let's getting back to what we have left. So step two, this folding and putting particular sugar. So after obtaining this oligosaccharide, now this is called what? Can you read this? Glycoprotein. Glyco means sugar, carbohydrate. So it is like a combined word, protein plus sugar, because this protein now has additional sugar. So this glycoprotein, so in other words, every protein made by this ribosome attached on the surface of a rough ER are literally every, are actually glycoproteins. Because there is no way you can escape this thing. Every protein will obtain their own particular type of the sugar once they are inside of this ER, rough ER. So it's like become interesting where all these proteins are shipped. Okay? Now, stage number 3 is what? What happened? The purpose, they are done here. They're finished. So these proteins, finished protein, must be shipped out of this rough RER. The question is how? The how is being answered in this part of the cartoon. Can you explain how by looking at this cartoon? So this is the final. This is the final version of the shipment, the vehicle, like a boat, ferry boat for this particular cargo. If we call this as a cargo, and this is a boat, ferry boat, how do you generate? How to generate this transport boat is illustrated here over here. So step 3, somehow in the process of generating this shipboat, and final version finished this ferry boat inside of which now the cargo is contained. So can you explain how to generate this ferry boat? One word. Can you describe one word How do you deal with this? Yes, pinch. This one will be pinched out. So this is what? Originally, this was a part of the membrane, areal membrane. And one edge of this membrane is pinched off to cut this thing. Now it becomes separate, independent vehicle. Okay. So how would you name this transport boat? Hmm? Is there any particular like a feeling preference for you to name this? This transport vesicle, because this is a vesicle. Now this vesicle has been generated by pinching of the membrane of RER. But one important thing is inside of this pinched of whatever the membrane, now separate membrane, you better have this finished process, the finished protein folded and carbohydrate added. Okay. By the way, adding the carbohydrate to the like a substance like protein, we call glycosylation. Another big word. Glycosylation, carbohydrate addition. So this protein is folded and glycosylated and now finished and enclosed within this transport vesicle and then shipped to the next generation. I mean, next destination. Okay, that's all you need to know about. And actually, if you understand all these things, and you are now way up here in terms of the level of understanding of this biology, as like in terms of you are at even the cell biology level. That's it. Okay, good. Now, the next destination is this Golgi apparatus. So they accept all those transport vesicles coming from the RER. Actually, here we can see the obvious benefit of depending this shipment transport on the transport vesicle. It's a very important thing. Like, let's talk about transport vesicle from. So transport vesicle in terms of like chemical nature, what is it? Chemistry wise, transport vesicle is membrane. Yeah, this is a cell membrane. Transport vesicle is made of phospholipid bilayer membrane, chemistry wise. So what's the beauty of it? Is there any benefit you can think of using this phospholipid bilayer as some kind of a shuttle vehicle between two locations, which are also bounded by cell membrane type? They can easily fuse. This is the word. Easily fuse together. You don't really have to do elaborate things, but once you have this transport vesicle, by bumping into the membrane, they become the integrated one. Like, let's try to imagine this is a transport vesicle, and you have cell membrane. that is coming into. I'm coming at you, and what's next? So, what's the next step? Can you imagine? If they bump each other, and it become like this, isn't it? So if you had this, you have something, cargo inside, what happened now? This cargo will be freely released into the inside of this. So if this is a transport vesicle, TV, and if this is a destination, which is Golgi, now this dumping this material inside into this inside of this Golgi would be like so easy. So that's the beauty of relying this membrane in terms of like a membrane bound something, some transport of vehicle in this delivery. There's a tremendous advantage. Okay? So Golgi can easily accept any kind of different cargoes coming from our yard. Fine. So what are you going to do with all those cargoes here? Because this Golgi is some kind of middle station. Golgi, another like a postal office, is behaving as if they are cellular postal office. All those mail delivery coming into this office, and then they have to sort. Okay, this particular one should go some, and another thing should go another location of the cell, type of this protein sorting. All those proteins destined for some other location of, like, for example, membrane or outside of cells are somehow correctly identified in the Golgi and then processed accordingly. How? It's amazing, isn't it? As if, like, if I compare this Golgi as a postal office, what would you have in the first place to make the function of this Golgi so efficient? If they behave as if a postal office, all those mails better have what? Intrinsically. When you mail something, you usually put particular code on your mail, isn't it? Zip code. And by this zip code, you don't even know what this zip code is. This is a zip code. So all those mails have this particular different zip code, so that postal office can correctly recognize, and then they efficiently sort them out and then deliver them. And so is the Golgi. Because originally these different proteins have their own different kind of a zip code have attached on the protein. So Golgi can recognize. It's an amazing thing. Golgi can recognize different protein zip code to distinguish, okay, this protein, this protein should go out of the cell. Oh, this protein should go to the cell membrane. Oh, this protein should go some other place. That's what we call this protein zip code. Different protein has different zip code. So even some protein ending up inside of the nucleus have their own unique zip code. Some protein should go to mitochondria, has their own unique zip code like this. And the good thing is Golgi can distinguish. Golgi can recognize and distinguish all these different things. So that's what like we say Golgi proteins function, Golgi apparatus function is to protein sorting. And another thing is continuing on this putting the carbohydrate, glycosylation. So I compare this the glycosylation occurring inside of the rough ER is something like a basic foundation cosmetic makeup. Then the further detailed, more sophisticated carbohydrate addition is being done in cerebral Golgi. So once they go through this proteins, all these proteins will have vastly different and variety of the different types of carbohydrate will have on their surface. So each different protein will show their true recognition marker as their true identity once you have this. So this is additional like extra gravy. I have already explained this. So what do you mean salt? Some protein will go into the cell membrane, some others will go even outside of the cell, or some others will go to the mysterious place like called the lysosome. So we are going to look at what's lysosome and what's their function in a minute shortly. So this is lysosome. The function of lysosome is like our like stomach in the cell. As we, as an entire one organism, we have a stomach functioning as a digestive like the place. Each different cell has their own small area where the cellular level digestion take place. So any waste material will be handled inside of this lysosome, including old DNA, old carbohydrate, old proteins, everything. And sometimes some captured bacteria come in, then they will be shipped into this lysosome, and lysosome will digest, degrade them. So usually if these cells are somehow, if we can capture all those coming infecting bacteria and then eat them, then we have no problem. We have no worry for this bacterial infection. But what kind of cell can eat bacteria in our body? Can you imagine that? Any cells that can eat bacteria? Are there any such thing like we have? Yes, we have. Yes. White blood cell. Some type of white blood cell can even eat incoming bacteria, and after eating them, all those eaten bacteria will be sent into lysosome and they were all digested. Good? Yes. Hmm? Yeah. T killer? What does it mean, T killer? A cytotoxic T cells is different one. White blood. There are several different white blood cells exist. The T cells, there are two different types of T cells, and the T killer cells are something really like bully cell. So whenever they something see unpleasant, they kill them. They puncture the cells, so all those cells can be punctured. And it doesn't only apply to the bacteria cell, but any cells. If this cytotoxic T killer cell doesn't really like any cells, and then it kills them. And usually the virus infected cells or the bacteria or even cancer cells can be the target of these T killer cells. Okay. But I'm talking about the other cells, eating cell. Okay. Actually, the name of this white blood cell specializing in eating is macrophage. And it has many arms extending and grab bacteria and then eat them. And Korean for macrophage is, yeah, you know what that is. So this is one such type of white blood cell. And the thing that they can do this is due to the presence of this lysosome. Everybody has a lysosome, but this macrophage has another utility, found another utility of their presence of this lysosome. And this is another very important thing about this autophage, self-eating. Phage is like now we have understand probably phage. Phage means something like eating something. So every lysosome is not only about, so this is nothing to do with the white blood cells, macrophage, but every different cell has this ability of something called autophage. Autophage means a self-eating. So usually what happens is they degrade their own materials, of course. That's like normal function of this lysosome. But especially it is kicked on under circumstances of like the brief period of starvation. Normally, lysosome will degrade their internal cellular material only if, maybe I should buy the second generation of this stupid camera. They are now actively on advertisement. This one is like, it's not listening to my order. So usually all the enough organelles, like especially mitochondria is the one such a particular target, and other materials on cellular, old material, especially in the case of this old mitochondria is very important because if mitochondria become too old, it can generate very dangerous chemical compound for us, which is like the trigger for the aging of the cellular aging. So we better take care of this old mitochondria very quickly. But anyway, but this autophage is not something like a normal, regular basis of degrading any waste product inside. But on the starvation condition, they actively digest some of the young, fresh ones, not waste products, but actively degrade their functioning, normally functioning, healthy organelles become the victim of this, what is called an autophage. It is some kind of brief temporary survival mechanism evolved. What are you going to do if there is no supply of food from outside? As an individual cell in your body, what are you going to do? Nobody has any promise anything. Okay, food will be shipped on like 2 days later, or so you hang in there for a moment. You don't receive any such message or promise. So under certain uncertain condition, what you can do, best what you can do is at least try to survive for another day or two. How? By self-destructing what you have, so that later, anticipating later the ration will come, then I will restore things. That's the principle of this autophage. And this is something like this, the periodic starvation, brief periodic starvation as a health benefit when you do this. So everybody, not everybody, a lot of people have this follow this practice of intermittent starvation. Intermittent fasting is probably the best, like correct word. Intermittent spelling is correct. I'm not sure, but in Korean, like Ramadan. Uh, so now everybody knows that this is really good for your health. And the mechanism by which you can have any health benefit is due to this. Like lysosome will degrade through this. If you go into this intentional starvation by triggering this intermittent fasting, then mitochondria, I mean, not mitochondria, your cell will interpret this as this like brief starvation. So like I will do this autophage and then actively degrade, especially some of those potentially problematic mitochondria-like stuff. So you can significantly reduce the potential risk of this, some risk coming from mitochondria. That's how you can increase your health benefit and as a result, consequently, your like longevity even. Because in these days, a lot of studies actually pointed that one of the main reasons why your cells has to be old is due to the old mitochondria. So if you take care of this, like actively, then you should be able to increase the chance of living much longer. Is this and healthier? But anyway, the mechanism by which a lysosome can do this digestion is the inside of, just like exactly like the chemical condition of our stomach. Inside of our stomach is highly acidic, isn't it? So all those foodstuff can be easily digested. Even, of course, you have also a lot of the specialized digestive enzyme inside of our stomach. And so is in the lysosome too. There are many different types of specialized digestive enzymes present, but the all common characteristic is they are highly activated only under specific acidic condition, just like those enzymes in our stomach too. Okay, but how do they maintain their inside condition so acidic? Is that same mechanism that keeps the inside of the stomach acidic. That's why we need to look at a little more closely about this, because once you understand this, then you can easily automatically understand the mechanism by which our inside of the stomach can become highly acidic. So this is it. By the way, this cartoon is drawn by myself using Adobe Illustrator. So this is my original illustration. I have a copyright. So it's joking. Copyright. I have no intention to claiming any copyright on this particular drawing. But anyway, I just try to brag myself that I made this. So altogether, hydrolase is what? Something that break down hydrolysis. So all different kind of enzymes are there. Like some are specialized in protein hydrolysis. Some others are specialized in carbohydrate. Some others are in lipid. So all kinds of different hydrolyses are here. But all the one common things about all these different hydrolyses are acidic. They can be active only under acidic condition like pH 5 and under. pH 5 is like probably upper limit of, but it can be much less than pH. So as you go down, it will become more acidic, isn't it? Like pH is an indication. pH is a parameter of how acidic it is. If you didn't know what the pH means, okay, just in case, pH, when you have pH 7, this is kind of neutral. If you go beyond pH 7, then this is alkaline. Alkaline, which means basic. If you go below pH 7, it will become acidic. So, for example, strong hydrochloric acid, which is extremely dangerous, nasty acid. The pH of hydrochloric acid is of something about pH 2. And the condition of highly active our stomach is also something like pH 2. And inside, the internal condition of this lysosome is pH 5 is very generous, probably much lower than pH 5, maybe even some 3 or 4 as well, be possibility. But anyway, that's not important. But the important thing about this is, by looking at this cartoon, can you understand how this lysosome create the acidic condition inside? First thing you have to understand is this, whether this acidity or like alkaline or basic condition is all kind of like the condition of dependent upon what? The pH is simply the parameter or indicative measure of how much hydrogen ion concentration you have. It's like it's a logarithm, so that's why the more you have like a stronger the concentration of hydrogen ion, you have a lower pH value, whereas the higher the pH value, you have much less hydrogen ion condition. In other words, acidic condition means stronger, higher hydrogen ion concentration. If you have more hydrogen ion, it will become acidic. It's all depend on the hydrogen ion. Now, can you? So in other words, it's not really that easy. I mean, it's not really that difficult to create acidic condition. It's just a matter of putting more hydrogen ion inside, accumulate the high hydrogen ion concentration inside, then it will become acidic. That's simple, isn't it? But how? How can you put more hydrogen ion inside? Transporter, but there's one catch. Like you have already high enough hydrogen already present, and probably the chances are outside the concentration of hydrogen will be less than accumulated highly concentrated area. But still, do you think you will be able to easily transport this hydrogen from outside to inside? Because according to the rule of like a nature, the material will always try to move from higher concentrated area to the lower concentrated area. That's why when you cigarette smoke, when you puff the smoke, that smoke will spread outside, because here this is a concentrated cigarette smoke, and then there is a much less concentrated area, then it will go from higher to lower concentration. That's the nature, the rule of nature. We call that as a diffusion. Every material, every molecule will follow this rule. From higher to lower concentration. But this is something totally opposite. You are trying the opposite. Whenever you're trying to something that opposite to the nature, what did I say? You have to open your wallet, which is ATP, right? So some transporters is capable of spending this money, the ATP, turn this ATP into ADP, which is ATP hydrolysis. And this is like energy required for this difficult reaction. And thanks to this, hydrogen can go inside. Without this, if you are not willing to spend any energy, reverse thing will happen. Hydrogen, because this area will have a much higher concentration than outside, so those hydrogen ion, if they have any chance, then it will go out of the inside to move outside. But this is the energy is the one that keeps still this thing inside by putting actively any outside hydrogen into the inside. That's the mechanism. And what did I say? The same mechanism is behind our stomach to make the condition of our stomach so acidic. Exactly the same mechanism. So the same transporter is also exists on the surface of our stomach, so membrane, and do exactly the same thing. So that's why it works to look at and understand this mechanism of lysosome maintaining the internal condition acidity. Okay? Now it's mitochondria. So that's something that before that, so we have looked at what? Smooth ER, rough ER, Golgi, and lysosome, and they are all the part of so-called endomembrane system. And except for the smooth ER, this rough ER, Golgi, and lysosomes are part of this, the transport of this material to the outside. From Golgi, there are basically three destinations for any given protein. Either you ship them into the cell membrane or outside, or you ship any particular other protein to the Golgi. To lysosome. Like for example, all these acid hydrolyses are actually coming from Golgi to lysosome. So if I ask, are all these acid hydrolyses are glycoproteins or not? What do you think? All these acid hydrolyses or whatever, there's several different kinds of enzyme proteins hydrolyzing. Are these glycoproteins too? Yes. Why? Yeah, they all went through, starting from rough ER to Golgi, two places where what? Glycosylation happens? Obviously. Let me ask you another thing. All those proteins on the cell membrane proteins, are all these membrane proteins glycoproteins too? What do you think? Like membrane proteins, what do you mean by membrane protein? What I mean by, if this is membrane and all these proteins embedded on the cell membrane, are they all glycoproteins or not? Yes. Everybody. Every membrane protein should be glycoprotein. Why? Because every one of these must have gone through this same route, starting from rough ER, Golgi to this. Another question. Are all those secreted protein that shipped even out of the cell, like insulin or any other, like what digestive enzyme and all those different kind of proteins, are they glycoproteins too? Answer is yes, because they are all have taken the same route as the all this transport protein that we have seen so far today. Okay? So that's the one part of this chapter. Now we can move into the mitochondria. And I've mentioned several times the like, function of importance of mitochondria because uh, they they generate a huge amount of ATP. But the particular cellular process, by which the ATP is produced in huge amount is we call cellular respiration. What do you know? In like a common the layman's term, what do you think of this cellular respiration? What is respiration to you? Respiration. What is it? Breathing. Is it? And what's this breathing got to do with this ATP generation? In other words, ATP generation is the job of mitochondria's metabolism, but to finish them, to achieve that without any problem, oxygen is essential. So for mitochondrial function for this, whatever, the ATP generation, the presence of oxygen is essential, as we will see later on. That's why to breathe, to have a supply of enough oxygen is extremely important for any heterotrophs. Any heterotrophs breathing, any heterotrophs of eukaryotes, having any cells having mitochondria, this oxygen is such an essential. And that is the reason why we have to die out of oxygen. If you cut off the supply of oxygen for 5 minutes, usually you die. Why? Why do you die if you don't have any oxygen supply for like 5 minutes or longer? Not every part of your body dies, but especially some very critical part of your body, like here and here, because these two areas is particularly notorious for high ATP demand. They need huge amount of ATP for their proper function. And to do so, the mitochondria of this area is constantly generate enough ATP, but without oxygen, they can't. So these two areas stop functioning. And unfortunately, as one typical multicellular organism, if either this or that doesn't function, then you are pronounced dead. No matter how other parts are still alive and functioning. So that's why. So that's the importance of another way of looking at the importance of mitochondria. Another peculiar thing about this mitochondria is, as we have seen in the previous lecture, they have their own DNAs. Oops. And cell division wise, like mitochondria divide. When cell divide, mitochondria also divide, but their multiplication, division, is totally independent from the host cells, so to speak, host cells' decision to divide or not. And even they have their own ribosomes inside. In other words, they keep their own copies of ribosome inside And if you think of, well, that could be, yeah, that's possible because ribosomes can be everywhere, right? So ribosome can be like anywhere, free living inside of the. By the way, do you know the general general internal space of a cell? Like, let's say this is a cell, eukaryotic cell. So this must be nucleus. Then what would you call the rest of the whole space here? Cytoplasm, right? Cytoplasm, right? Generally. So ribosome can be anywhere, like they can be everywhere, as we have seen, like in the previous lecture, like in the first session. Ribosome can be anywhere in the cytoplasm, or some other ribosome can be specifically attached on the surface of ER. Fine. So by the same token, why not ribosome can go inside of this mitochondria, and it should be nothing spectacular or something special. Yeah, they can go inside of mitochondria, but why do we make this thing? Why do we bring up this thing and then make us something like particularly some special about this? You will see. So, first off, mitochondria is weird in that, unlike other regular organelle, it consists of double membrane. It's not something like a lipid, phospholipid bilayer. We have two phospholipid bilayers. One layer we have an inside internal membrane. We have an outside membrane. Two cell membrane like stuff. Double membrane structure. Inner membrane is an extensive folded structure called the cristae. It's not really important, whatever. Uh, of course, it has its own importance, but we are not going to pay any special attention. So this is a typical drawing of mitochondria. So this one is an external membrane, and this is an internal membrane which is folded extensively. Okay. If you have this extensive folded one, obviously it will generate much more surface area. Oh, that's a good thing, but whatever. We don't. The important thing is they have several copies of identical DNAs. And this DNA's special thing is, unlike DNA residing in the nucleus, this DNA, hello, this DNA is circular, and this circular DNA is what? Hallmark of, characteristic of prokaryotic DNA. All the prokaryotic cells, bacterial cells and all the other prokaryotes have this circular DNA, whereas eukaryotic DNA is a linear DNA. That's the big difference. This really distinctive difference between eukaryote and prokaryote. And this mitochondria has a prokaryotic DNA. Oops. When somebody said that mitochondria also have DNA, then probably some of you might probably reply that, yeah, why not? Because some part of some fragment of DNA could contaminate, go into the mitochondria. So what's wrong with that? It's possible. But then when you realize that the DNA nature is totally different, like prokaryotic versus eukaryotic, then you have to think twice. Oops, what's going on over here? And so now we are ready to go over that endosymbiosis theory. That theory was formulated to explain the origin of mitochondria and the chloroplast. Jesus Christ. Siri responded. So is it a good thing or bad thing? Siri misunderstood. Did I say anything something similar to "Hey Siri"? I don't believe so. Jesus. Okay. This is, as I mentioned briefly earlier. So this is something about independently living bacteria. So in the case of mitochondria, it's got to be an aerobic bacteria, which means oxygen requiring bacteria. Aerobic means oxygen requiring, which is like characteristic of this metabolism based upon this ATP synthesis. So mitochondria has, if we consider mitochondria as a like ancestor of mitochondria at least, as a free living independent bacteria, that must have acquired some very unusual, unique, new metabolism, which is oxygen requiring but producing huge amount of ATP. It's a whole new metabolic ability. This is goody. This is really goody. And somehow, unfortunately, this one was internalized by a bigger host cell. And then, instead of destroying this, probably for the sake of a host cell, it would have been much more beneficial. If the mitochondria was instantly destroyed and then become a lunch, well, then that was a one-day lunch. But I don't know for some reason, and I don't know how can that be possible. I cannot really think of, but maybe some mutation must have happened to the host cell. So not being able to destroy this incoming mitochondria and somehow let it go inside, let it live freely together. And so I just jokingly said they signed the contract so that we live together peacefully ever since. And we have some of a mutual benefit, obviously, of course. But anyway, whatever happened, mitochondria survived inside and then able to multiply independently and then kept their integrity by keeping their own genes inside. So they're living. And something also happened. So that was one event. But then later, another such lightning struck twice to the same person. Or you won the lottery twice. What's the possibility of winning the lottery twice in your lifetime? But it happened. So another similar independent living organism entered, named chloroplast. This one has totally new ability, being able to do this photosynthesis. So those who have hit twice like this became the ancestor of plants, whereas those who unfortunately have just only one event of hitting this lottery became the animal. That's the what endosymbiosis theory states. So this, in your textbook, this is an illustration of the endosymbiosis theory. So here, by the time this entry of mitochondria, this cell has almost achieved like eukaryotic features. One thing missing is the energy provider factory. But fortunately, this guy came in. Wow. So it became living together, so animal cell. But then to some of this, the second event of intruder, this time around, the chloroplast, showing totally new ability of photosynthesis. Now you can produce your own carbohydrates, like became autotroph. So that's the whole story goes. But it's all fairy tale. What evidence do you have? One evidence is the DNA. which is circular, which means prokaryotic DNA. Okay. Another strong evidence is ribosomes also. I mentioned that they have also their own ribosome, and then I just cynically mentioned the possibility of cytoplasmic ribosome can come into, but that is not the case because the ribosome is totally different. Like DNA, the ribosome also, we can distinguish the prokaryotic ribosome from the eukaryotic ribosome, and this ribosome existing inside of the mitochondria and chloroplast are exactly those prokaryotic bacterial type. In other words, they can show off total independence. They can have their own genes, and they have this gene activity, and their gene product can be translated, made, converted into protein by their own ribosome. So I don't need you if I want to, for example. I can live totally independent outside of you is probably kind of message. Of course, these days, they totally outside of the host cell mitochondria, no single mitochondrion or chloroplast can live outside of a host cell in these days because they have already accustomed and then adapted to this symbiotic relationship. But anyway, so to speak, theoretically, that's the thing. The third evidence is this. Two double membrane things. Mitochondria and chloroplast, both of them possess this double dual membrane system, and this outside and inside, outer and inner membranes, chemical structure is different. That's a very weird thing. Outer membrane is chemically very similar to that of prokaryotic, but another group of prokaryotic archaebacteria, whereas inner membrane is more similar to, chemically speaking, to the modern bacteria. What's the meaning of this? Let's say this is mitochondria coming in. This one is a bigger prokaryote wanting to become first eukaryote. We all know that our direct ancestor, our old eukaryote, direct ancestor of the eukaryote is one particular branch of archaebacteria. Right. That is a really like fact. And if this thing has happened, see what happened. If this thing just simply coming in, it would have included the part of this outside membrane. So that's how they obtained dual membrane. And if we examine this dual membrane, chemically speaking, that was something like original the archaebacteria, and inside one is similar to the modern day bacteria, is something another evidence of this entry things. So these three, like DNA, circular DNA, their own, and prokaryotic ribosome, and finally this dual membrane. Outer one is that of archaea, and inner one is that of modern bacteria. Is that the three kind of a solid evidences that support the endosymbiosis theory. Okay, so cytoskeleton is too lengthy, so I don't have any intention to go any further today. So hopefully, although it's too late, but I hope you can enjoy the rest of the Friday night. So that